Good morning, good afternoon, good evening from wherever you're watching from. Welcome back to the Public Health Circle YouTube channel, where we learn all matters public health. Thank you for taking your time to watch this video. Epidemiology is the study of the determinants, occurrence, and distribution of health and disease in a defined population. Infection is the replication of organisms in host tissue, which may cause disease. A carrier is an individual with no overt disease who harbors infectious organisms. Dissemination is the spread of the organism in the environment. Epidemiology utilizes an organized approach to problem solving by first, confirming the existence of an epidemic and verifying the diagnosis. Second, developing a case definition and collating data on cases. Third, analyzing data by time, place, and person. Fourth, developing a hypothesis. Fifth, conducting further studies if necessary. Sixth, developing and implementing control and prevention measures. Seventh, preparing and distributing a public report. And finally, evaluating control and preventive measures. Chain of infection is the sequence of events required for an infectious disease to spread, involving an infectious agent reservoir portal of exit mode of transmission portal of entry and a susceptible host. There are three major links in disease occurrence. 1. The etiologic agent. 2. The method of transmission, which can be by contact, by a common vehicle, or via air or a vector. And 3. The host. The etiologic agent is the causative agent of an infection, like any microorganism that can cause an infection. Now, let's learn about the factors to consider when describing the agent. Pathogenicity. The pathogenicity of an agent is its ability to cause disease. Pathogenicity is further characterized by describing the organism's virulence and invasiveness. Virulence refers to the severity of an infection, which can be expressed by describing the morbidity, or incidence of disease, and mortality or death rate of the infection. An example of a highly virulent organism is Yersinia pestis, the agent of plague, which almost always causes severe disease in the susceptible host. The invasiveness of an organism refers to its ability to invade tissue. Vibrio cholerae organisms are non-invasive, causing symptoms by releasing into the intestinal canal an exotoxin that acts on the tissues. In contrast, Shigella organisms in the intestinal canal are invasive and migrate into the tissue. No microorganism is assuredly a virulent. An organism may have very low virulence, but if the host is highly susceptible, as when therapeutically immunosuppressed, infection with that organism may cause disease. For example, the poliomyelitis virus used in oral polio vaccine is highly attenuated and thus has low virulence, but in some highly susceptible individuals it may cause paralytic disease. The infecting dose, which is the number of organisms necessary to cause disease, varies according to several factors. These include the organism itself, the method of transmission, the site of entrance of the organism into the host host's defenses, and the host species. Another agent factor is specificity. Some agents, for example Salmonella typhimurium, can infect a broad range of hosts, while others have a narrow range of hosts. Staph, for example, infects only humans. Other agent factors include antigenic composition, which can vary within a species, such as in the influenza virus or Streptococcus species. Additionally, antibiotic sensitivity, resistance transfer plasmids, and enzyme production are important factors to consider. The reservoir of an organism is the site where it resides, metabolizes, and multiplies. The source of the organism is the site from which it is transmitted to a susceptible host, either directly or indirectly through an intermediary object. The reservoir and source can be different. For example, the reservoir for S. typhi could be the gallbladder of an infected individual, but the source for transmission might be food contaminated by the carrier. The reservoir and source can also be the same, as in an individual who is a permanent nasal carrier of S. aureus, and who disseminates organisms from this site. The distinction can be important when considering where to apply control measures. The method of transmission is the means by which the agent goes from the source to the host. The four major methods of transmission are by contact, by common vehicle, by air, or via a vector. In contact transmission, the agent is spread directly, indirectly, or by airborne droplets. Direct contact transmission takes place when organisms are transmitted directly from the source to the susceptible host without involving an intermediate object. This is also referred to as person-to-person transmission. An example is the transmission of hepatitis, a virus from one individual to another by hand contact. Indirect transmission occurs when the organisms are transmitted from a source, either animate or inanimate, to a host by means of an inanimate object. An example is transmission of Pseudomonas organisms from one individual to another by means of a shaving brush. Droplet spread refers to organisms that travel through the air very short distances, that is, less than 3 feet from a source to a host. Therefore, the organisms are not airborne in the true sense. An example of a disease that may be spread by droplets is measles. Common vehicle transmission refers to agents transmitted by a common inanimate vehicle, with multiple cases resulting from such exposure. This category includes diseases in which food or water as well as drugs and parenteral fluids are the vehicles of infection. Examples include foodborne salmonellosis, waterborne shigellosis, and bacteremia resulting from use of intravenous fluids contaminated with a gram-negative organism. The third method of transmission, airborne transmission, refers to infections spread by droplet nuclei or dust. To be truly airborne, the particles should travel more than 3 feet through the air from the source to the host. Droplet nuclei are the residue from the evaporation of fluid from droplets, are light enough to be transmitted more than 3 feet from the source, and may remain airborne for prolonged periods. Tuberculosis is primarily an airborne disease. The source may be a coughing patient who creates aerosols of droplet nuclei that contain tubercle bacilli. Infectious agents may be contained in dust particles, which may become resuspended and transmitted to hosts. An example occurred in an outbreak of salmonellosis in a newborn nursery in which salmonella-contaminated dust in a vacuum cleaner bag was resuspended when the equipment was used repeatedly, resulting in infections among the newborns. The fourth method of transmission is vector-borne transmission, in which arthropods are the vectors. Vector transmission may be external or internal. External or mechanical transmission occurs when organisms are carried mechanically on the vector. For example, Salmonella organisms that contaminate the legs of flies. Internal transmission occurs when the organisms are carried within the vector. If the pathogen is not changed by its carriage within the vector, the carriage is called harborage, as when a flea ingests plague bacilli from an infected individual or animal and contaminates a susceptible host when it feeds again. The organism is not changed while in the flea. The other form of internal transmission is called biologic. In this form, the organism is changed biologically during its passage through the vector, for example, malaria parasites in the mosquito vector. An infectious agent may be transmitted by more than one route. For example, Salmonella may be transmitted by a common vehicle, such as food, or by contact spread, like a human carrier. Francisella tularensis may be transmitted by any of the 4 routes. The third link in the chain of infection is the host. A host is an organism that harbors an infection. The organism may enter the host through the skin, mucous membranes, lungs, gastrointestinal tract, or genitourinary tract, and it may enter fetuses through the placenta. The resulting disease often reflects the point of entrance, but not always. Meningococci that enter the host through the mucous membranes may nonetheless cause meningitis. Development of disease in a host reflects agent characteristics and is influenced by host defense mechanisms, which may be nonspecific or specific. Nonspecific defense mechanisms include the skin mucous membranes, secretions, excretions, enzymes, the inflammatory response, genetic factors, hormones, nutrition, behavioral patterns, and the presence of other diseases. Specific defense mechanisms or immunity may be natural, resulting from exposure to the infectious agent, or artificial, resulting from active or passive immunization. Epidemiologic methods are scientific approaches used to investigate and analyze the distribution and determinants of health and disease in populations. The three major epidemiologic techniques are descriptive, organizing data by time, place, and person; analytic, incorporating a case control or cohort study; or experimental. Although all three can be used in investigating the occurrence of disease, the method used most is descriptive epidemiology. Once the basic epidemiology of a disease has been described, specific analytic methods can be used to study the disease further, and a specific experimental approach can be developed to test a hypothesis. In descriptive epidemiology, data that describe the occurrence of the disease is collected by various methods from all relevant sources. The data are then collated by time, place, and person. A description of epidemiologic data by place must consider three different sites: where the individual was when disease occurred, where the individual was when he or she became infected from the source, and where the source became infected with the etiologic agent. Therefore, in an outbreak of food poisoning, the host may become clinically ill at home from food eaten in a restaurant. The vehicle may have been undercooked chicken, which became infected on a poultry farm. These differences are important to consider in attempting to prevent additional cases. All pertinent characteristics should be noted: age, sex, occupation, personal habits, socioeconomic status, immunization history, presence of underlying disease, and other data. Once the descriptive epidemiologic data have been analyzed, the features of the epidemic should be clear enough that additional areas for investigation are apparent. The second epidemiologic method is analytic epidemiology, which analyzes disease determinants for possible causal relations. The two main analytic methods are the case control or case comparison method and the cohort method. The case control method starts with the effect, which is the disease, and retrospectively investigates the cause that led to the effect. The case group consists of individuals with the disease, while a comparison group has members similar to those of the case group except for the absence of the disease. These two groups are then compared to determine differences that would explain the occurrence of the disease. An example of a case control study is selecting individuals with meningococcal meningitis and a comparison group matched for age, sex, socioeconomic status, and residence, but without the disease, to see what factors may have influenced the occurrence in the group that developed disease. The second analytic approach is the cohort method, which prospectively studies two populations: one that has had contact with the suspected causal factor under study and a similar group that has had no contact with the factor. When both groups are observed, the effect of the factor should become apparent. An example of a cohort approach is to observe two similar groups of people, one composed of individuals who received blood transfusions and the other of persons who did not. The occurrence of hepatitis prospectively in both groups permits one to make an association between blood transfusions and hepatitis. That is, if the transfused blood was contaminated with hepatitis B virus, the recipient cohort should have a higher incidence of hepatitis than the non-transfused cohort. The case control approach is relatively easy to conduct, can be completed in a shorter period than the cohort approach, and is inexpensive and reproducible. However, bias may be introduced in selecting the two groups. It may be difficult to exclude subclinical cases from the comparison group, and a patient's recall of past events may be faulty. The advantages of a cohort study are the accuracy of collected data and the ability to make a direct estimate of the disease risk resulting from factor contact. However, cohort studies take longer and are more expensive to conduct. Another analytic method is the cross-sectional study, in which a population is surveyed over a limited period to determine the relationship between a disease and variables present at the same time that may influence its occurrence. The third epidemiologic method is the experimental approach. A hypothesis is developed and an experimental model is constructed in which one or more selected factors are manipulated. The effect of the manipulation will either confirm or disprove the hypothesis. An example is the evaluation of the effect of a new drug on a disease. A group of people with the disease is identified, and some members are randomly selected to receive the drug. If the only difference between the two is use of the drug, the clinical differences between the groups should reflect the effectiveness of the drug. An epidemic investigation describes the factors relevant to an outbreak of disease. Once the circumstances related to the occurrence of disease are defined, appropriate control and prevention measures can be identified. In an epidemic investigation, data are collected, collated according to time, place, and person, and analyzed and inferences are drawn. We have more detailed videos about disease outbreak investigation. Please watch for a better understanding of the entire process. Thank you for watching. Please like this video and share it with your friends and colleagues. Don't forget to subscribe to this channel for similar informational videos. Bye!